This tutorial describes running the sample clustering pipeline on the KnowEnG platform. You start by uploading and selecting a genotype file, such as a spreadsheet of samples describing non-heterogeneous somatic mutations. When the pipeline runs, it can identify clusters of samples that have similar genomic signatures, such as cancer patient subtypes. Each of these gene signatures are available as part of the pipeline results. The results download includes a spreadsheet of the top genes per cluster with their scores. Choosing to enable the knowledge network can help mitigate issues of very sparse or heterogeneous data. It does this by broadening the somatic mutation observations across gene neighbors through known interaction relationships. If you also have phenotypic descriptions for each sample, this pipeline can identify phenotypes that are highly correlated with each cluster. These data relationships are available to explore in the visualization view of results, which we cover in detail later in this video. Let's get started running this pipeline. Launching the KnowEnG platform will take you to the home screen of the application. Here you have access to new additional information about the app, as well as buttons to get you started running a pipeline. We'll click on Start a New Pipeline. This takes us to the home screen for the pipelines. As new pipelines are developed, they will be added to this screen. You can learn more about each pipeline by clicking on the name and viewing the contextual information in the panel on the right. This info panel is also available throughout the workflow to provide information relevant for each step. For now, we'll click on Sample Clustering Pipeline, and then click on Start Pipeline to begin. The first thing we do in this step is to make sure our species selection is correct. For this demo, we'll be using cancer datasets, so let's keep the default setting of human. Since there's no preloaded spreadsheets to choose from in this demo, we're going to first upload our genomic features file. We will be using a dataset from TCGA that contains the gene-level non-synonymous somatic mutations from 381 lung adenocarcinoma patient samples. Click on the Upload New Data button at the top right of the table. The upload controls will appear. Here you can drag a file into this field or click on Browse. We'll click on Browse and then navigate to the file on our system. Our file should begin loading in the data table with a progress indicator. Once the file is uploaded, click on the checkbox to the right side of the row to choose the file. Click on the Next button at the bottom right of the screen to move to the next step. This step is optional, but recommended if you have phenotype data for your samples. The file we'll be using contains values for 10 different phenotypes, observed for the same patient samples contained in the lung adenocarcinoma dataset. Examples of these phenotypes include cancer stage, days of survival, and smoking status. Click on the Upload New Data button at the top right of the table. Again, click on Browse and we'll navigate to the file on our system. Your file should show up in the data table with uploading progress info. Once it's done uploading, click or hover on the row and then click on the Select button to choose it for this pipeline run. Click on the Next button at the bottom right of the screen to move to the next step. On this page, use the default final clustering algorithm, k-means. In the number of clusters returned text box, enter 6. More information on choosing the number of clusters can be found in the help text contained in the info panel. For now, we'll click Next. This step in the pipeline setup is where we decide whether to use the knowledge network in our analysis. The somatic mutation profiles we uploaded for this example, like some other types of genomic data, are difficult to cluster using traditional techniques for two important reasons. First, the gene features describing each sample are extremely sparse. Samples typically have only a few dozen mutations in the entire exome. Second, they are very heterogeneous. Two phenotypically similar samples might have only a single mutation in common. Using the knowledge network mitigates both of these difficulties by diffusing the somatic mutation observations across gene neighbors through known interaction relationships. So we'll select yes to use the knowledge network and then select the Human Net Integrated Network. Check out the info panel for more information about selecting a network to use. Keep the network smoothing at its default settings and click Next. This page gives us the option to use bootstrapping. Bootstrapping uses repeated runs of the analysis on random subsets of the original data to find and better quantify a more robust solution. Select Yes for using bootstrapping and keep the default settings for the rest of the parameters and click Next. The last step in the pipeline setup displays a summary of parameters that were chosen for this particular run. The system generates a default job name, but we can change this name here. Since the same job can be repeatedly run with only slightly different parameters, you may want to attach a note to the job to help document your process. To access this feature, click on the Notes pencil icon. After we've reviewed our selections, we'll go ahead and click Submit Job. Once the job is submitted, a new page will display the status of the run and should say Running. From this page, you can navigate to the results page or start a new pipeline. Leaving the screen will not cancel your running job. To see your results, click Go to Results page or Results in the main navigation. We'll click on Go to Results page. On the results page, you should now see a listing of all jobs you've run, with the latest one at the top. In our case, we are only seeing the job we have just submitted. Wait for a few minutes until the status column for this entry turns to a green check mark. Then click on the job name and look at the info panel on the right. Here you can access all of the information about this run, including data files used, parameters, notes, and favorite status. In this panel, you are also able to download, delete, or favorite a result. To open the result, double-click on the row in the table or click on the View Results button in the info panel on the right. Because we chose to use bootstrapping, the heat map displays the results of the consensus clustering. The rows represent the samples in our data, now grouped into clusters. The columns also represent the samples and appear in the same order as the rows. The cell in a given row and column represents how often the row sample and the column sample were assigned to the same cluster during the bootstrapping process. This gives us a way to assess the quality of the cluster assignments. The cluster sizes are reported in the table on the right. You may use the Add Phenotypes to Compare feature under the heat map to add sample phenotypes to the visualization. Each phenotype is visualized using a mini heat map that is aligned with the main heat map above and represents the same samples in the same order. A p-value of significance is on the left, and the heat map color legend is on the right of the mini heat map. Click on the legend to display the values for the phenotype. With these mini heat maps, we can visually assess whether patterns in the values correspond to the clusters in the heat map. To download the results, click on Results at the top of the page. Click on the latest job and then click the download icon in the right panel to download the zip archive. In the downloaded archive, you will have the sample to cluster ID mapping located in the file samplelabelsbycluster.txt, the consensus matrix values in the consensusmatrix.txt file, and a file mapping the input gene IDs to their internal knowing identifiers located in genemap.txt. There is also a file called topgenesbycluster.txt that is a binary valued spreadsheet with gene names as rows and cluster IDs as columns that indicate if a gene was in the top 500 important genes for the cluster. Information about other files included in the download archive is included in the Quick Start Guide for this pipeline. This demo was created using a beta version of the platform. The website display and URL may not exactly reflect what you see on your screen. We hope this tutorial has been helpful and that KnowEnG is a useful tool for your research. This tutorial only contained one set of choices for the sample clustering pipeline. We suggest you try changing various parameters to see their effects. More information on these options is provided on the info panel on each page of the pipeline setup. Additional information about the KnowEnG platform is available in the support section of the application and on our website, knoweng.org. Thank you.